Scientific Communications by Lyndal Neilsen. GABA is the primary inhibitory neurotransmitter known to counterbalance the action of the excitatory neurotransmitter glutamate. Nearly half of all the synapses in the brain have some sort of GABA receptor. The neurotransmitter begins developing in the embryonic phase and continues throughout life. During the central nervous system development in the human body, GABA changes the main role from being excitatory transmitter to an inhibitory transmitter, which is caused by an influx of calcium into the postsynaptic neuron, which is originated from the release of GABA. This switch in development is influenced by the neuronal chloride concentration. When the chloride levels are high, GABA acts as an excitatory neurotransmitter. However, when the levels are lower, GABA acts more inhibitory. There are two types of GABA receptors, GABA A, which is ionotropic, and GABA B, which is metabotropic. GABA A is a ligand chloride channel that opens and closes to allow for ions to flow into the neuron, making the neuron more negative. GABA B receptors are found both pre- and postsynaptically. For the activation of potassium and calcium channels are coupled together via G-protein pathway. G-protein regulates voltage-gated calcium channels and affects cellular processes such as trafficking and desensitization. Benzodiazepam is one of the most successful drugs available since the revolution in the 1950s. The drug is commonly known as Valium. Valium has the power to treat many central nervous system disorders, including epilepsy, anxiety, reducing muscle spasms, alcohol withdrawal, and seizures. Benzodiazepam increases neuronal chloride ion influx upon binding with GABA, resulting in a depressive response. There is a high risk of becoming dependent on the drug, leading to substance abuse. Benzodiazepam dependency risk factors may be misuse, driving impairment, motility due to overuse, and being taken with alcohol. Alcohol and Valium both affect GABA receptors in the brain. When Valium is taken with alcohol, there is a greater percentage of the drug reaching the brain, therefore creating a more intoxicating effect. Risk factors include drowsiness, confusion, depressed feeling, slow motor function, laboured or slow breathing, poor coordination, or death. Benzodiazepam has a very short peak effect, which means it's quick to take influence in the body. Following quick symptom relief, the drug has a long half-life that ensures for a gradual decline in levels so there is lower withdrawal symptoms. Benzodiazepam's peak onset is generally 1 hour with a half-life metabolite of between 3 and 100 hours with a 5 mg dose. Moving on to chloride channels. Chloride channels are involved in a diverse range of biological functions, including epithelial fluid secretion, cell volume regulation, and smooth muscle contraction. Mutations of the chloride channels can result in several human diseases, including cystic fibrosis, macular degeneration, hyperplexia, and kidney stones. Around 70 to 85% of resting membrane conductance is carried by chloride ions. Cellular homeostasis maintains a balance of many factors that contribute to keeping a cell healthy. When the membrane potential is depolarized, opening the in-strength channels of the GABA A receptor will hyperpolarize the membrane and vice versa. So when the membrane potential is hyperpolarized, opening will become depolarized. Moving on to channelopathies. Channelopathies are a heterogeneous group of disorders resulting from the dysfunction of ion channels located in the membranes of all cells and many cellular organelles. Channelopathies that affect skeletal muscles include disorders from myotonia to flaccid paralysis. CLC1 channels stabilize resting membrane potential and help membrane repolarization in skeletal muscle cells. Functional loss of CLC1 channels reduces the influx of chloride that is required to compensate for depolarization. This can then lead to spontaneous firing of action potentials and have slower repolarizing rate. Thank you for listening to my video.